Hello, dear students. Today, we will explore the fascinating world of structural and numerical chromosomal mutations. We will be focusing on key types such as deletion, duplication, inversion, translocation, aneuploidy, and polyploidy. All of these play critical roles in both genetic disorders and evolutionary biology. So, if I simplify the objectives, understanding the structure, organization, and species specific nature of chromosomes and their fundamental role in heredity, evolution, and biodiversity. My students will explain the types, causes, and biological impacts of structural and numerical chromosomal mutations. That will include their molecular mechanism during cell division. We'll also identify examples of genetic disorders and we will evaluate the effects of chromosomal aberrations on health, adaptation, and speciation in various organisms. We'll assess the application and implications of chromosomal mutations in agriculture, genetic counseling, and reproductive health. And yes, we'll highlight their advantages and disadvantages. So, if I start with what exactly a chromosome is. So, what are these chromosomes? Chromosomes are long, thread like structures. They are composed of DNA and associated proteins, serving as the fundamental carrier of genetic information in all living organisms. In eukaryotes like me and I, chromosomes adopt a compact three-dimensional organization that enables large DNA molecules to be efficiently packed within the tiny cell nucleus. So, this compactness is crucial for the regulation of gene expression and it ensures the accurate distribution of genetic material during cell division. Chromosome number refers to the total count of chromosomes present in the nucleus of a cell. It is a species specific characteristic, meaning each species has a fixed number of chromosomes. In humans like us, the diploid number is 46, that is 23 pairs, with one set inherited from each parent. Chromosomes are dynamic structures. Their shape and size vary across different stages of cell cycle, reflecting their complex role in cellular function. So, in eukaryotic cells, DNA is packaged with histone and other chaperone proteins into chromatin, whereas prokaryotes usually contain a single circular chromosome without such organized packaging. So, any change in the structure or number of chromosomes can lead to significant alterations in an organism's phenotype. And these are the changes we call chromosomal mutations, or, dear students, chromosomal aberrations. So, chromosomal mutations differ from gene mutations in that their effect is large and can often be observed microscopically. These mutations are generally classified into two major categories: the structural mutation, which involves alteration in the physical arrangement of chromosome segments, and numerical mutations, which involve changes in the number of individual chromosomes or entire chromosome set. While many chromosomal mutations lead to genetic disorders or diseases, not all are harmful. Some occur in non coding regions of DNA and may have little to no functional impact. However, even such mutations can contribute to evolutionary variation, potentially offering adaptive advantages in certain environments, dear students. So, if I take an example, we see chromosomal rearrangement has played a significant role in the diversification of plant species. So, in conclusion, I can say chromosomal mutations, whether structural or numerical, are major contributors to genetic diversity. That's how you and I are different. This study not only enhances our understanding of heredity and evolution, but are also important implications for diagnosing and managing genetic disorders. Now let's start with the structural and numerical chromosomal mutations. Structural chromosomal mutations refer to large-scale alterations in the physical structure of chromosomes. These mutations typically arise due to errors during cell division, especially in the processes like crossing over, where homologous chromosomes may pair abnormally. Chromosomes naturally have the ability to bend, loop, and form sticky ends, making them prone to breakage and incorrect reattachment. As a result, segments of chromosomes may be lost, duplicated, inverted, or incorrectly relocated. These mutations often happen when chromosomes do not align or separate properly. Gene segments are broken or misplaced. Genetic material is inserted into the wrong chromosome, or sometimes whole segment of genes are lost or repeated. And if I classify them broadly, they're categorized into 4 types. Number 1, deletion. A segment of the chromosome is missing or lost, leading to loss of genetic material. Number 2, duplication. A segment is copied and inserted again, so it will increase gene dosage. Number 3, inversion. A chromosome segment breaks and reinserts in the reverse direction. And last but not least is translocation. A segment is transferred to a non-homologous chromosome, either reciprocally or non-reciprocally, dear students. So all of these structural mutations, they can disrupt gene function. They can disrupt gene expression, potentially causing all the genetic disorders. Even developmental issues we see in some people, they contribute to evolutionary changes. Yes, while some are harmless or even beneficial, many structural mutations can result in serious biological consequences, depending on their location and size. So let's start with elaborating deletion. Chromosomal deletions are a type of structural mutations involving the loss of a segment of a chromosome. And these changes are often irreversible, as the missing DNA is usually degraded and cannot be replaced. Deletions can vary in size from a small section of DNA to large portion of chromosomes, and they have significant biological consequences. Yes, depending on the region and genes. Deletions typically result from chromosome breakage caused by factors such as radiation, certain chemicals, viral infections, transposable elements, or errors during recombination or DNA replication. So these chromosome segments that lag during anaphase or are incorrectly repaired can be digested by nucleases, of course, leading to permanent loss. So there are 2 major types of deletions. Number 1 is terminal deletion, where a single break leads to the loss of an end portion of the chromosome. Number 2, intercalary or interstitial deletion, which results from 2 breaks that eliminate a central segment of the chromosome. These deletions can be detected cytologically during meiosis, often appearing as deficiency loops during chromosomal pairing at the patch-teen stage. While homozygous deletions are often lethal, heterozygous deletions can also produce harmful effects, depending upon the gene content loss. So several human genetic disorders are associated with deletions. So human genetic disorders, if I name them, which are caused by deletion, we have Cry-Duchat syndrome. We see deletion in the short arm of chromosome number 5. Duchenne muscular dystrophy or DiGeorge syndrome. Detection techniques for deletion, my dear students, include karyotyping. You can detect large chromosomal changes. FISH, fluorescence in situ hybridization, uses fluorescent probes for locating specific DNA sequences. and comparative genomic hybridization, or CGH we call it. It identifies copy number variation. And last but not the least, we see PCR, the polymerase chain reaction, which detects smaller deletions at the molecular level. Deletions can disrupt many genes and pathways, potentially causing developmental, physical, and neurological disorders. Now, if I see the variations in chromosome structure, we talk about duplication. Duplication is a type of structural and chromosomal mutation where a segment of chromosome is copied one or more times, resulting in extra genetic material. This causes certain genes to exist in more than two copies, which can lead to imbalances in gene expression, developmental abnormalities, or evolutionary advantages. There are several types of duplication based on the location, orientation of the duplicated segment. Number one is tandem duplication. The copied segment is placed adjacent to the original one and in the same order, dear students. But in reverse tandem, we see duplicated segment is adjacent but in reverse orientation. Third one is displaced duplication. The duplicated segment is present elsewhere in the same chromosome. And last, we see transposed duplication. The duplicated segment is inserted into a non-homologous chromosome. There are some extra chromosomal duplications as well. If the duplicated region contains a centromere, it may exist as a separate chromosome. If we talk about examples, we see bar eye phenotype in Drosophila melanogaster, fruit fly, caused by duplication of the 16th A segment on the X chromosome, leading to narrow, slit-like eyes. Or 22q11.2 duplication syndrome in humans, caused by an extra copy or part of chromosome 22. We see MECP2 duplication syndrome affecting intellectual development, primarily in male counterparts. We see Charcot-Marie-Tooth disease type 1 resulting from duplication on chromosome 17. So, cytological examination of chromosomes showed that bar allele results from duplication of a small segment, that is 16A of the X chromosome. Now, moving on to inversion. Inversion is a type of structural chromosomal mutation in which a segment of a chromosome breaks off and rotates 180 degrees and reinserts into the same chromosome, but in reverse order. Although the total genetic content remains unchanged, the rearranged sequence can disrupt gene function or regulation, especially if vital genes or regulatory regions are affected. So there are two major types of inversion. Paracentric inversion, the inverted segment does not include centromere, or pericentric, the inverted segment includes the centromere. So inversions are usually caused by errors during DNA repair, cell division, or exposure to mutagens like radiations, chemicals. In heterozygous individuals, inversion loops may form during meiosis to allow pairing of homologous chromosomes. If crossing over occurs within these loops, it can result in duplication, deletion. While most inversions are harmless and may not lead to visible changes in the phenotype, especially in homozygotes, they can occasionally lead to genetic disorders due to breakpoint effects. When a gene is disrupted at the inversion site, or position effects students when gene expression is altered due to new locations. For example, in the insect Culepa frigida, inversion contributes to phenotypic diversity and evolutionary fitness. In humans, some cases of infertility, miscarriages, or disorders like hemophilia have been linked to chromosomal inversions. Another well-known example is chromosome 12 inversion, linked to extreme height in certain individuals. They are taller than normal. Now moving on to translocation. Translocation is a structural chromosomal mutation where a segment of one chromosome breaks off and attaches to a non-homologous chromosome. Unlike deletions or duplications, translocations do not change the total amount of genetic material, but they rearrange the genetic sequence, which can affect gene function and regulation. There are three main types of translocation. Number one is simple translocation. A single segment from one chromosome attaches to the end of a non-homologous chromosome. Number two is shift translocation. A broken segment is inserted into the internal region, not at the end, of a non-homologous chromosome. Third one is reciprocal translocation. Segment from two homologous or non-homologous chromosomes are exchanged. And this is called illegitimate crossing over and is often balanced, meaning no genetic material is lost or even gained. We see a special kind of reciprocal translocation, which is called Robertsonian translocation, which occurs between two acrocentric chromosomes. Chromosomes with very short arms, such as chromosome 13, 14, 15, and 21. In this case as well, the long arms of these chromosomes fuse and the short arms are lost. This type of mutation is responsible for familial Down syndrome, where chromosome 21 attaches to chromosome 40. Though balanced, translocations may not cause immediate problems, they can lead to unbalanced gametes, resulting in genetic disorders like infertility, miscarriage, or developmental abnormalities in offsprings. Some forms of leukemia and lymphoma are also caused by translocations. We see that in between chromosome 15 and 17 in acute promyelocytic leukemia. Examples in plants include Nithera, where all 7 chromosomes are involved in reciprocal translocation. They form a ring-like structure during meiosis. And this translocation plays a huge role in evolution and speciation. Now let's move on to numerical chromosomal mutations. These mutations involve changes in the number of chromosomes in a cell. Number one is aneuploidy. It's a type of numerical chromosomal mutation that results from addition or loss of one or more individual chromosomes, leading to a chromosome number different from the typical diploid set. It is a form of heteroploidy, where the chromosome is altered without affecting the entire set. Aneuploidy is broadly classified into 2 types. Hyperploidy, addition of chromosomes to the diploid set. You see trisomy, presence of an extra chromosome, like we see in Down syndrome, the trisomy of chromosome number 21, or tetrasomy, 2 extra chromosomes of the same type. Double trisomy, we refer it as like 2n plus 1 plus 1, or pentasomy, or other forms found in plants like wheat and datura. Hyperploidy, loss of 2 chromosomes from diploid set. Monosomy, loss of a chromosome, like we see in Turner syndrome, we see monosomy of chromosome X. Nullisomy, loss of a homologous chromosome pair, and it's usually lethal. Double and triple monosomy are also observed in species like maize. Aneuploidy typically arises due to non-disjunction during meiosis or mitosis, where chromosomes fail to segregate properly. This causes gametes or cells with abnormal chromosome numbers. While plants often do not survive such mutations, animals may survive with developmental abnormalities, infertility, or mental retardation, depending on the affected chromosome. Dear students, common examples include Down syndrome, caused by the non-disjunction of chromosome 21, Turner syndrome, resulting from absence of one X chromosome in females, The aneuploidy we're talking about plays a significant role in genetic disorders, in cancer, and evolution. And yes, you can detect it through karyotyping, FISH, or molecular techniques. Now, moving on to polyploidy. Such numerical chromosomal mutations where whole genome is multiplied. Polyploidy is a form of euploidy, a chromosomal mutation where an organism contains more than two sets of chromosomes. In diploid organisms, chromosomes occur in pairs. However, in polyploid organisms, the basic set of chromosome is multiplied to form triploid, tetraploid, pentaploid, hexa, octa, all of these structures. While polyploidy is rare in animals, it's widespread and significant in plants. It plays a major role in plant evolution, in speciation, and their students in crop improvement. We see major three types of polyploidy. Autopolyploidy is the number one. Autopolyploids contain multiple chromosome sets. They originate from a single species. The genome is simply multiplied without hybridization. We see autotriploids. There are three sets of chromosomes. These are usually sterile due to irregular meiotic divisions, leading to unbalanced gametes. Despite sterility, they are agriculturally valuable. Seedless fruits such as bananas, watermelons, tomatoes, and apples are often autotriploids. Natural autotriploids like dubgrass, cynodon dactyl, they're also common autotetraploids. They have four sets of chromosomes and are usually fertile. These autotetraploids can arise naturally or be artificially induced using colchicine. It's a chemical that inhibits spindle fiber formation. Yes, during the cell division. Examples include potato, alfalfa, coffee, and grapes. Autopolyploids tend to show gigantism, larger cell size, increased water content, and higher biomass. Their student's number two is allopolyploidy. Allopolyploids are formed by hybridization between two different species, followed by chromosome doubling to restore fertility. Since the original hybrid is often sterile due to the lack of homologous pairing, artificial doubling using colchicine can restore their fertility. A classic example is Raphanea brassica, a hybrid between reddish Raphanus sativus and cabbage, Brassica oleracea, created by Russian geneticist G.D. Karpenchenko in 1927. Though he expected a hybrid with edible roots and leaves, the plant showed cabbage roots and reddish leaves opposite to his goal. Another important example is triticale, the first successful man-made cereal crop, created by crossing wheat and rye. Triticale exists in multiple ploidy levels. Tetraploid, cross between diploid wheat and rye. Hexaploid, cross between tetraploid wheat and rye. Or octoploid, triticale cross between hexaploid wheat and rye. Hexaploid triticale combines the high protein content of wheat with the high lysine content of rye, making it nutritionally superior. Third one is autoallopolyploidy. Autoallopolyploids contain genome sets from multiple species as well as repetitions of the same genome. They are rare but can occur naturally or be developed through complex breeding techniques. Let's move to the significance of polyploidy. It's a major evolutionary force in plants and a powerful tool in crop improvement. Polyploids often exhibit larger organs, greater environmental adaptability, and higher resistance to the disease than their diploid counterparts. Many ornamental plants are autotetraploids, valued for their larger flowers and longer blooming periods. Triploid crops are preferred for seedlessness, dear students, and high sugar content, like in bananas and sugar beets. Moving on to chromosomal mutations and diversity appearance. Chromosomal mutations are often what makes us unique as living organisms. Physical features such as blue eyes, blonde hair, and even freckles were a result of chromosomal mutations, allowing humans to become more diverse in their appearance. Hair color, eye color, beauty spots, and other features caused by these genetic mutations all promote variation among the human race. Applications of chromosomal mutations. Number one is gene mapping and research. Scientists use chromosomal mutation to rearrange genes in specific ways. This helps in studying how genes work and solving various biological problems. Medical applications are also there. In medicine, understanding chromosomal mutation helps doctors diagnose and treat genetic and hereditary disorders. In some cases, these mutations can even be used in molecular therapies to manage diseases. Plant and animal breeding is the third one. Chromosomal mutations are useful in improving plants and animals. They help breeders develop new varieties with better traits, such as higher yield, disease resistance, or faster growth. Last but not the least is evolutionary studies. These mutations are important in the study of evolution. They help scientists understand how the structure and number of chromosomes have changed over time in different species. We also see production of seedless fruits. Induced polyploidy helps in increasing the number of chromosome sets and is used to grow seedless fruits and vegetables like bananas and watermelons. We also see genetic experiments. Monoploids with a single set of chromosome are very useful in genetic research. Since they have only one copy of each gene, scientists can easily study gene function and mutations. Dear students, let's move to advantages of all these chromosomal mutations. When we hear the word mutation, we often think of harmful effects or disease. However, not all mutations are bad. Some chromosomal mutations can actually be helpful to organisms, supporting their survival, diversity, and evolution. We see survival advantage. Certain chromosomal mutations can help organisms survive better in their environment. These beneficial changes are especially important when environmental conditions change or when new challenges arise. For example, lactose tolerance. It's a mutation that allows adults to digest milk. In populations that depend on animals like cows and goats for milk, this was a big survival advantage. Some people carry genetic mutations that offer resistance to diseases or HIV immunity due to specific genetic changes. Or skill cell mutation, which gives resistance to malaria in affected individuals. A height-related mutation found in tribe in Ecuador is linked to a lower risk of cancer. So these are the examples which show how mutations can protect individuals from illness and help them adapt to their environments. We also see genetic and physical diversity. Chromosomal mutations play a major role in creating diversity among living organisms. Like harmless mutations gives rise to features, different eye colors, blue, brown, green, various hair colors, blonde, black, red, or you see freckles and beauty spots. This diversity not only makes individuals unique, but it also strengthens a population by increasing the chances of survival under different environmental conditions. So, my dear students, in summary, chromosomal mutations are not always harmful. Some provide real advantages that improve survival, that promote evolution, and creates the rich diversity we see in the natural world today. Yes, there are disadvantages of chromosomal mutations. While some chromosomal mutations can be beneficial, many mutations can be harmful to health and survival of an organism. Chromosomal abnormalities and human genetic disorders, which are caused by chromosomal mutations, include trisomy of chromosome number 21, Down syndrome, which causes developmental delays and distinctive facial features. We see trisomy 18 and 30. They lead to serious health problems and shorter life expectancy. Klinefelter syndrome, it's an XXY condition which affects males. XYY syndrome may result in taller than average height and learning difficulties. We see Turner syndrome, X0, which affects females, causing short height and infertility. Or triple X syndrome affects females, often without major symptoms, but sometimes with learning challenges. Other health conditions and diseases of humans are some chromosomal mutations can increase the risk of developing non-inherited diseases, including cancer, caused by mutations that lead to uncontrolled cell growth. We see that in lung cancer, breast, or blood cells. Diabetes, asthma, and sickle cell anemia can also be influenced by chromosomal or genetic mutations. For example, sickle cell anemia is common among individuals of African descent and affects red blood cell shape and oxygen transport. Now, different approaches to reduce the risk of chromosomal aberrations. Although chromosomal aberrations cannot always be prevented, certain steps can help lower the risk, especially before and during pregnancy. Parents can reduce the chance of chromosomal abnormalities in their children. How? By maintaining good nutrition and a balanced diet, avoiding harmful substances like tobacco, alcohol, and drugs, taking prenatal vitamins, especially folic acid, before and during pregnancy, visiting a doctor before conceiving for a health checkup, getting genetic counseling, especially if there is a family history of genetic disorders. The risk of chromosomal abnormalities increases when a pregnant individual is over 35 years of age. In such cases, healthcare professionals may recommend additional tests or assisted reproductive technologies to reduce risks. Now, impact of chromosomal mutations on genetic counseling. Genetic counseling is a process that provides information and support to individuals or families who may be at risk of genetic disorders. It helps them make informed decisions about reproduction, medical care, and lifestyle choices. Chromosomal mutations can affect fertility, pregnancy outcomes, and the livelihood of passing genetic conditions to future generations. Understanding their impact is essential in modern healthcare and reproductive planning. Now, if we see the risk assessment, one of the key roles of genetic counseling is to evaluate the risk of inherited chromosomal abnormalities in a family. Certain chromosomal mutations, such as translocation, inversion, or aneuploidies, like Down syndrome, Turner syndrome, these may be inherited or occur de novo. A trained genetic counselor studies family history, medical records, and genetic test results to identify individuals or couples at risk. For example, if one parent is a carrier of a balanced translocation, where no genetic material is lost or even gained, there is a change or chance that their child could inherit an unbalanced form, which might cause physical or mental disability. Diagnostic testing advances in cytogenetics have enabled the detection of chromosomal mutations using precise diagnostic tools, which are crucial in genetic counseling. Some commonly used ones are karyotyping, which allows visualization of all chromosomes under microscope to detect abnormalities such as deletion duplication. We have FISH, fluorescence in situ hybridization, which helps detect smaller or more subtle chromosomal changes by using fluorescent probes that bind to specific DNA sequences. We see chromosomal microarray analysis, which detects copy number variation that may not be visible in karyotypes. These tests can be conducted before or during pregnancy to determine if the fetus has any chromosomal abnormalities. Family planning and reproductive choices are also important. Couples with a known risk of chromosomal mutation often face important decisions regarding reproduction. Genetic counseling supports them by discussing possible options, such as pre-implantation genetic diagnosis, used with IVF to screen embryos for genetic defects before implantation. Prenatal testing, such as amniocentesis or chorionic villus sampling, which analyze fetal cells for chromosomal abnormalities. Assisted reproductive technologies may be recommended to increase the chances of a healthy pregnancy, especially for individuals with chromosomal rearrangements. Adoption or use of donor gametes may also be considered if the risk of passing on a disorder is high. So, my dear students, for biology students, grasping the types, causes, and consequences of chromosomal mutations, both structural and numerical, is crucial for careers in genetic medicine and biotechnology. Remember, while these mutations can lead to developmental abnormalities and diseases, they also drive evolution and have transformative application in crop improvement and species diversification. Therefore, studying chromosomal mutations bridges human health and agricultural innovation, underscoring their vital role in modern biology. The students hope this lecture was really helpful to you, and we will be providing more lectures in the upcoming times. Thank you.